It looks like we've got in the 80s right now.
Oh, very good. Okay.
Yep.
Thank you. Thank you very much. Okay, so I'll be talking to you about what I would call the greenest turf insect and beech leaf disease management. And let's see, here we go. And so a little bit of an overview of my presentation is that there's going to be a shift in how pesticides are used for managing white grubs. That's for those groundskeepers who use conventional insecticides. And that will have implications for chinch bug management, although chinch bug management could be of concern for all turf managers. And so I'll present a green approach for managing all turf pests. And then for the second half of my presentation, I'll be going over beech leaf disease management, especially with very new information that provides really practical information for those who wish to manage that disease. So 2025 was really quite an interesting year in that the Connecticut State Legislature passed and the governor signed into law a bill that specifically bans the use of neonicotinoid insecticides on turf by October 1st of 2027. And that has implications for conventional turf managers. And the reason for this ban really stems from the fact that U.S. Environmental Protection Agency really goofed when they did the initial registration for imidacloprid and other neonicotinoids used in turf, um, in that the aquatic toxicity data that they used to allow the registration and that kind of shaped the use patterns that were allowed used this organism, which is Daphnia magna, or the water flea, as a representative of aquatic invertebrates. Unfortunately, this particular organism has an LC50, that should be LC50 for lethal concentration, a median lethal concentration of 85 parts per million, which is about 2,000 times less sensitive than the closely related shrimps, for which the LC50 is 35 parts per billion. And after reviewing aquatic organism toxicity data, US EPA in 2017, they changed their level of concern for chronic exposure of aquatic organisms from the previous level, which was 1 part per billion, to only 10 parts per trillion. And so that's 100-fold reduction in the permissible exposure of imidacloprid in aquatic systems. There was a paper published in 2024 by U.S. Geological Survey scientists that did a nationwide survey of aquatic systems to find out what pesticides they would find in those water bodies. And of several compounds of concern, they really highlighted the presence of imidacloprid, bifenthrin, and chlordane, which is a holdover from use back 50 years ago, as posing the greatest risks to aquatic organisms. And so if you look at their statistical analysis of those compounds that are likely impacting the biodiversity of our aquatic systems, imidacloprid was contributing about 14% of the variation that could be attributed to reducing the number of species found in streams. They really focused on what we call EPT organisms. That's Ephemeroptera, Plecoptera, and Trichoptera, which are respectively mayflies, stoneflies, and caddisflies, which are very sensitive organisms to pollutants. And these are very important organisms because they serve as the base of the food chain. And if you lose those organisms, then you probably lose the ability to grow fish as well in those streams. And really, the sensitivity of these organisms is quite extraordinary. If you look at this graph on the x-axis, you see the concentration of imidacloprid in nanograms per liter, which would be parts per trillion. And the species diversity, this is a count of numbers of species found in individual bodies of water. And you see that when you go from basically 1 part per trillion, where you have a larger number of species, by the time you get up as low as 5 parts per trillion, there's a sudden drop off that there are no streams that have the same number of species as you would have in the less contaminated streams. So both in the US and in Europe, it's generally recognized that there's a threshold of probably around 6 to 10 parts per trillion of contamination in the water of neonicotinoids that leads to the loss of our most sensitive species to that type of pollution. And to put it in perspective, it would only take about a 7-inch square, or about 1/3 of a square foot, of the amount of imidacloprid applied to turf to control white grubs. If that were to contaminate a body of water, it would contaminate an Olympic pool volume of water to a concentration of 10 parts per trillion. So that's really quite a small amount of area ending up contaminating water to be of potential concern to survival of our most sensitive aquatic organisms. And this is a real concern, as portrayed by this graph. There is a spike in the concentration of imidacloprid found in waters, and I believe these are data from Connecticut that showed that the level of concentration was somewhere around between 40 and 50 parts per trillion, which is clearly above the threshold level of concern, which is where that line is right there. And turf, applications to turf covering larger areas and with potential for example, granules or overspray going onto hardscape, washing off into storm drains would be the principal way in which the active ingredients could end up in aquatic systems. So turf was specifically implicated as being the likely source of pollution that could lead to loss of biodiversity in our streams. Well, the reasons why imidacloprid are applied to turf are principally to manage white grubs, which are the larvae of these various species of scarab beetles. If we start from the upper left, we have Asiatic garden beetle. Upper right is European chafer. Lower right, Oriental beetle, and lower left is Japanese beetle. Now, we're not so concerned about the adults of these species, and unless you want to protect flowers from the feeding by adults. Rather, they're the larvae that we're really most concerned about. And the reason for the orange arrows is that those species are not really well controlled with imidacloprid anyway. So it's always been true that we only got about 50% kill of Asiatic garden beetle with imidacloprid right at the get-go. And starting about 2013, Oriental beetle, we were having control failures with imidacloprid. And in turf in Connecticut, certainly Oriental beetle is the most important of these 4 species. So imidacloprid was no longer an appropriate tool to use to manage white grubs anyway. So another interesting thing is that you could substitute a celloprene if you're using conventional chemistry to control white grubs. But if we use a celloprene to control white grubs and you have it properly incorporated into the root zone of turf with rainfall or irrigation, you end up with essentially a total elimination of white grubs for about 2 years, possibly longer. And that should not be the goal for white grub management, because if we wish to have a biologically viable system and natural regulation of pest populations, we need to maintain very low populations to at least provide an opportunity for natural enemies to be able to survive in that environment. And so we do have parasitoids that attack Japanese beetles. And then there are, of course, very common ants that are found in turf that are very effective predators that will eat the white grub eggs, as well as rove beetles and ground beetles. And then there are various diseases as well, insect pathogenic nematodes, bacteria, and fungi, that if we don't have any hosts there, then those agents will not be able to maintain their presence in a turf system. So one of the really important tools to consider for control of white grubs, rather than the synthetic chemicals, would be grub gone, which is Bacillus thuringiensis gallerii, and that would be applied at the time that you have uh, any time that you have larvae present in the soil. So that would go from the first week of August right up through to the time that the larvae are digging deeper into the soil, which might be usually would be around the first week of October. Another choice would be to consider using insect pathogenic nematodes. In either of these cases, if you were to use these tools, they would have to be applied in a manner in which they would become incorporated into the soil in the root zone through either significant irrigation or rain events. I wanted to draw your attention to this slide because insect pathogenic nematodes are not used very much for control of white grubs, but they have tremendous potential for biological control of white grubs, in particular the species Steinonema scarabaei, which you see on the left. The species of nematodes that are infecting the grubs can be identified by the color that those larvae turn. So if you see them turn this sort of reddish brown color on the right, that means that the grub was infected by a Heterorhabditis species. The larvae that are infected with Steinonema scarabaei turn a really distinctive yellow, sort of a mustard, a greenish yellow color. And I would offer to everybody attending this seminar that if you ever find any larvae, white grub larvae, that have this distinctive color, call me immediately because I would like to do more biological work with this nematode in trials that my colleague Albrecht Koppenhofer has conducted at Rutgers, and he's the person who described this species of nematode. Applications of this nematode usually result in close to 100% mortality of grubs in the affected turf. And so use of being able to figure out how to propagate this nematode and using it in turf could provide us the most effective tool if we could grow these nematodes. The chief drawbacks to using biologicals for curative control of white grubs is that the products need to be incorporated with rainfall to target first or second instars usually before damage occurs. And with climate change, unfortunately, we end up with a lot of periods of drought or with saturated soil, which would prevent proper incorporation of these materials. The other thing about using these biologicals is that they tend to be rather expensive, much more expensive than most of the chemical control agents. But that is really quite readily mitigated if you're only doing spot application to those parts of those areas in turf that have populations of white grubs that are above an economic threshold requiring some sort of treatment. So here's an interesting observation. Acelepryn is going to be the conventional insecticide that takes the place of neonicotinoids for control of white grubs. And also the biological agents that we use to suppress white grub populations don't control chinch bugs. So we can anticipate that turf management programs that are using these alternative materials will end up with more chinch bug problems. The old neonicotinoid based white grub management programs never had chinch bug problems because the neonics are rather effective at suppressing chinch bug populations. So here's the concern: hairy chinch bug. It's not controlled with many of the chemicals that would be used now for control of white grubs or biologicals. So we can expect outbreaks of chinch bugs to be more likely. Here's the life cycle of the hairy chinch bug, in which there's an egg, 5 nymphal stages. It has incomplete metamorphosis. And then the adults can take one of two different types of forms, either with fully developed legs, wings, or they may be brachypterous, meaning that they have short wings that are not functional for flying. The image on the left shows a area of turf that's heavily affected by chinch bugs. On the right, it shows that it shows the life cycle over the year, and there are 2 generations of chinch bugs per year. You can identify chinch bug problems by poking through the turf and looking for the bugs scurrying about, or you can use a coffee can method, which I'll show very shortly. A lot of turf managers may misidentify summer patch as being chinch bug damage because the two things look so similar. If there's summer patch, obviously applying insecticides won't help you. Application of a fungicide heritage June 15th and July 15th is a conventional fungicide method for managing this disease. But there is another approach that would be to replant with turf-type tall fescue or perennial ryegrass. And I'll go into the importance of those particular species of turf in a moment. The way that you can positively identify summer patch is to pull up some grass, wash off the roots, and look for these brown or black roots that are decaying, and that's a sign of summer patch. So to monitor chinch bugs, the standard methods are to use a coffee can. You cut out both ends of the can, pound it down into the turf, and then flood it with water. Or you can use a golf course cup changer, remove a plug of sod and put it into a bucket of water. And in both cases, the chinch bugs that are present in the turf, probably down in the thatch layer, will come floating up to the surface, and you can count them to see whether you are exceeding an economic threshold in which you might want to consider some sort of a pesticide treatment. Now, the conventional pesticides that have been used to manage chinch bugs would involve pyrethroids or neonicotinoids, both of which are now we recognize special concern for potential toxicity to aquatic organisms. There are low environmental impact tools that might be useful: Bovaria bassiana, insecticidal soap, horticultural oil, or azadirachtin products, none of which have a reputation for being particularly effective against chinch bug populations. There are reasons to think that Bovaria bassiana could be useful, and there are commercially available products that contain this insect pathogenic fungus. There are commonly outbreaks of or epizootics of natural infections of Bovaria fungus in chinch bug populations. And one suggestion has been that if you have good irrigation practices to keep moisture in the thatch area, that you should be able to induce greater suppression of chinch bug populations through natural infections with this fungus. That has not really held up to scientific study and trials to be a practical way to manage chinch bugs. On the other hand, the places where chinch bugs tend to be a problem usually are the drier areas of lawns, sunny, drier locations. And so that does suggest that there may be some there may be legs to the idea that Bovaria suppresses the chinch bug populations. Overall, the most important tool that we have available to manage all above ground insect pests and turf would be to plant high endophyte turf. Endophytes are fungi that are growing within the grass, or perhaps other plants also, um, that as they grow, they're secreting into the plant very highly toxic alkaloids. They're actually producing their own insecticides that make the above ground part of the grass blades not only unpalatable, but poisonous to anything that tries to feed on that grass. And so if you overseed with high endophyte grass varieties, which would be tall fescue, turf type tall fescue, or perennial ryegrass, then you will no longer have problems with chinch bugs. So you'll also no longer have problems with sod webworms, cutworms, army worms, bill bugs, um, yeah, and that basically covers a whole list of above ground insect pests. So it's basically one-stop shopping. You do overseeding with these high endophyte grasses, and you resolve the problem for all above ground insect pests feeding on that turf. You do not want to overseed areas of lawns on which your customers might want to allow their horse or cow to graze, because these high endophyte grasses were discovered by USDA because of their toxic effects on farm animals. And so it's very important for you not to be using high endophyte grass where people might be allowing their horse to graze. Now, the only way to know that you have high endophyte grass is to ask your seed provider. Sometimes even those bags of grass seed will not indicate on them whether they contain endophytes or not. And the reason for that is in order to claim that they are high endophyte, samples of grass seed have to be tested on a routine basis and checked to see that the fungus has remained viable within the seeds. If you do plant turf type tall fescue or perennial ryegrass, that will also get rid of the potential for problems with summer patch. So the bottom line here is establishing endophytic turf. It will manage all above ground turf insect pests. Now, if you were using conventional insecticides for white grub control and you combine high endophyte turf with chlorantraniliprole, that will provide a complete insect pest management for turf. An application of chlorantraniliprole or acelepryn prior to a rain event will provide typically about 2 years worth of control of white grubs with very little concern about toxicity to mammals or bees, because acelepryn is essentially non-toxic to honeybees and bumblebees. But the caveat there is that the same problems with application of pesticides to turf, with overspray reaching hardscape and then washing down nearby storm drains, is a concern when this product, acelepryn, is applied to turf, because it has long residual activity and high toxicity to aquatic organisms, just like the neonicotinoids. And so turf managers really have to up their game and make sure that they never allow the products that they apply to contaminate, have the potential to contaminate aquatic systems. Grubgon is a satisfactory alternative to chlorantraniliprole. So when you look at a completely green program for managing turf insect pests, you could use a high endophyte turf and apply Grubgon for control of white grubs. And in my opinion, that should be perfectly adequate for all turf insect pests. Okay, now for the second part of my talk, which is discussing beech leaf disease. Beech leaf disease is caused by a foliar nematode, Lidolenchus crenate, which infects the leaves when the leaves are very tiny and held within the buds during the fall, winter, and early spring. So the nematodes feed within the buds during the fall, winter, and spring, and they're feeding external to the leaf tissue that's developing. The nematodes are secreting a substance that will cause the leaves to change the way that they grow so that the cells are oversized. There are additional layers of cells. When those leaves then emerge, you can see that dark band between the veins. Those dark bands are very significant for the life cycle of the foliar nematodes because I suspect that the nematode requires these oversized or hyperplastic cells in order to colonize those leaves as they emerge from the bud. And so the only way that the nematode can then have the second part of its life cycle, which is not outside of the bud and within the leaf, is to have modified the way that the leaf grows so that it can invade those oversized cells. Now, I'm going to call these dark bands between the veins galls because of the way that they're formed. So there are two phases within the life cycle of beech leaf disease. At this time of year, they're feeding within the buds, and then during the summer, they're feeding within expanded leaves. All the damage that's important to the beech leaves is taking place only within the buds. Of course, it's important part of the life cycle that they're reproducing and reaching extraordinary numbers within the expanded leaves as well. The thing that I'd like to share with you today is that potassium phosphate products are really valuable for managing beech leaf disease, and in my opinion, are a standalone treatment for management of this disease. And the first hint that it might be useful was published in 2007 from a researcher looking at the impact of potassium phosphate applied to grains. I think it was wheat and two different species of nematodes. These nematodes are effective in infecting the wheat by causing the cells to change with their feeding. They form specialized structures that are essential for the nematode success. And what's curious about this is that the application of potassium phosphate prevented the change in the way that those cells were developing, thereby preventing the nematodes from having those specialized structures in which they could feed. So you see that there's a parallel here when we apply potassium phosphate that can prevent those specialized structures from being formed within the buds. My colleague Dan Herms, who's the vice president for research at Davey Tree, was the first person to look at potassium phosphate and its usefulness for protecting against beech leaf disease. So a little bit of history of beech leaf disease and his work. These slides are from Dan Herms that I'm sharing. 2012, it was first detected in Lake County, Ohio, and Cleveland Metro Parks is what CM stands for. They found it on their properties 2014, and then they started surveys 2016, and then they teamed up with Davey in 2017. So that's only 9 years ago that they started doing some trials looking at potassium phosphate. In that work, Davey Tree really focused on using something called polyphosphate 30, which is sold as a plant food. And I have to say here that this is not the product that you should be using for protecting your beech trees because it is sold as a fertilizer and does not have a pesticide label. Um, we should remember that any product that's applied to mitigate problems with pests must have a pesticide label. And there are plenty of products that we have available that have a label that allow us to use it for management of beech leaf disease. But in any case, they use this particular product in their research trials with Cleveland Metro Parks. And so what they did is that they had 20 treated trees, 20 untreated controls. Because this product is very systemic, they severed the root grafts that might be between these trees. They only used 2 to 4 inch diameter trees so that they could follow the populations in the leaves much easier. They could reach the foliage on those trees very readily. They did 2 applications with soil injections, May to June, and then again in August. And then they evaluated the trees every year. Dan Herms is an expert on emerald ash borer, and so he was very familiar with using canopy cover and canopy transparency for measuring the health of the trees. And they also measured fine twig dieback um, to see how well the product protected the health of the trees. Here are some of their data in which they looked at the percent of canopy cover that remained healthy. And so for the crown transparency, they had relatively healthy crowns when they used the potassium phosphate over the course of a 5-year period of time, whereas the canopies became very thin, very little canopy left for those untreated control trees. Another way of looking at these data was in the survival of the branches, and where they had used the potassium phosphate, the branches remained alive, whereas for the untreated checks, the control group is the salmon color. There was quite considerable loss in the number of twigs and branches. You can see in this photograph, these photographs, the comparison between the foliage on the treated group versus the untreated trees. Basically, what's amazing about this experiment is that the trees in this experiment had small trunk diameter, which means that their canopy was exposed to um, basically all those nematodes that were washing off from the beech foliage from the canopy above them on the larger trees. So they were always being subjected to colonization by nematodes in the late summer and fall, and yet the phosphate treatments were protecting the buds from being damaged by those nematodes that had entered the buds. So they did samples of the leaves to do nematode extractions. And the bottom line here is that when you look at the treated trees, the number of nematodes that they could extract from the leaves was something like 15 to 95% reduced compared to the untreated checks. So the potassium phosphate has been very effective in protecting the health of beech foliage in those situations where the trees were reinfected by nematodes every year. So the soil phosphate applications can maintain the canopy health of saplings with beech leaf disease and substantially decreases the foliar nematode populations. Now, I was aware of his work, and I went out to a meeting in 2023. And when I came back, I was talking to a number of arborists about the value of using potassium phosphate. And a number of arborists came to me independently and said, well, we've been treating our beech trees to protect them from bleeding canker, which is a Phytophthora disease. We've been treating every year with potassium phosphate to protect the beech trees from this other disease, and our trees are perfectly healthy, whereas surrounding beech trees that aren't treated have disease. So what it made me recognize is that there was potential even for much larger trees than in Dan Herm's experiments to be protected if you apply potassium phosphate on an annual basis. And the reason for this is rather interesting, is that potassium phosphate is readily absorbed into the plants. It can be applied through soil application, such as what Dan had done. You can do a bark spray, or you can do trunk injection of the bark, and you can do a foliar application. In any case, any way that you apply it to the plant, it will be readily absorbed and then will translocate both upwards and downwards. The way that it works is that it turns on what's called the systemic acquired resistance pathways in plants, which are generalized pathways that defend against various diseases. So, surprisingly, the same mechanism is involved for protecting a beech tree against infection by Phytophthora pini, which is what causes bleeding canker, and also against the foliar nematode. Um, Once you get the active ingredient into the tree, it cannot be metabolized. It's the active ingredient, the phosphite, is stuck inside the tree. Now, you might lose some of the material when the leaves fall during the during the autumn. However, it moves both upwards and downwards in the plants in the same direction as carbohydrates. So much of the product that reaches the foliage probably moves back down again, back down to the roots. And there would be dilution with new growth. So you do need to consider application every year with when you're using potassium phosphate. So my interpretation of what these arborists had observed were that multiple years of application will build up the concentration of phosphite in tissues until it exceeds a threshold that provides complete protection from nematodes in the buds. Really good evidence of this is this particular case. The gentleman in this image, Paul Quick, is not a leprechaun. That is a very large tree, 88-inch DBH tree in Darien, Connecticut. It had bleeding canker 2014, and Paul did applications, 2 applications per year as a bark spray, basal bark spray, every year since. And I'll show you a picture of the foliage on that tree. It's a little bit out of focus, but you can get the point here. These are leaves that are shaded, and I hope you can see my pointer here. Let's see, select a pointer tool. Okay. When you look at this particular leaf, you can see the dark bands between the veins. That is a shaded leaf in the under canopy of the tree, which when you think about it, when you treat a tree with a systemic material, shaded leaves will be conducting photosynthesis much less than those leaves exposed to the full sun. And so they will be accumulating much less of products that are dissolved in the xylem and carried in the sap. Now, if you look at the leaves that are more exposed to the sun, you see that they're healthy and they have no bands between those veins. And in fact, some of them even have signs of what I suspect to be a hypersensitive response. The plant responded to the presence of nematodes within the buds by actually killing some of the plant tissue and preventing the nematodes from being able to colonize those tissues. This is a pattern that I see in looking at the variation of susceptibility to beech leaf disease of various species of beech. American beech is highly susceptible, fairly uniformly highly susceptible. European beech vary in their susceptibility. Here's one cultivar called reversii, in which there appears to be a strong hypersensitive response from exposure to beech leaf disease within the buds. So I suggested in 2024 that people use the label directions for application of potassium phosphite, use it in a bark spray. And when you do this application, you do not need to include a penetrating organosilicone surfactant like Pentrabark. That has been known for let's see now, 35 or 40 years, that it does not enhance the absorption of products through the bark. And you could spray as high as you can comfortably reach to wet the bark without spraying the foliage, because this one-to-one dilution of potassium phosphate would be quite phytotoxic if it were sprayed directly to foliage. The results were quite dramatic. An arborist named Barry Dolby shared with me these images, and they're quite striking. In 1 year, he went from this particular tree being infected with beech leaf disease to being nearly perfectly protected. And he was able to obtain, with the 2 applications with a bark spray, he actually exceeded the concentration that really probably ought to have been the target, because here we can see a little bit of marginal necrosis, which is a salt burn, but otherwise the foliage is very healthy and functioning. The other sign of having exceeded an ideal dosage is that when you activate the SAR pathway in plants, it can prevent those plants from being able to defend themselves from mites. In this case, it is an Arenium mite, or an Eriophyte mite, that is a specialist on beech. And there were these inconsequential galls on the leaves caused by Arenium mites. On these trees, and I looked at them in the fall when the foliage was changing color, I found the same pattern, that the leaves that were on the underside of the tree and shaded did not receive as high a dose of the potassium phosphate as those leaves that were on the outer shell of the tree that would have been conducting more photosynthesis. And so if you were to use this approach of a bark spray, you might want to go back in with a foliar spray with the labeled rate, which is a very low concentration of potassium phosphate, to kind of touch up the foliage that is shaded, so that you can get an effective dose of phosphate within those parts of the tree. So potassium phosphate is very readily absorbed into the plants in any way that we apply them. A bark spray is probably the easiest application. You mix the product one-to-one with water. You don't need to add a penetrating surfactant. You should make 2 applications 1 month apart, late May and June, or June and July. You don't want to wait until after July to apply the product, because at that time of year, we can anticipate that the active ingredient would be moving downwards in the plant, and you would not be able to see benefits from a late season application until probably about a year and a half after having made that application. Trunk injection can also work with phosphites. These are data shared with me from Don Grossman from ArborJet. I'm not going to go over the data very much, other than to show you that the number of nematodes per gram of buds was basically reduced dramatically by having done trunk injections the previous growing season with potassium phosphate. And the rating of the foliage quality was greatly improved by having injected the trees with the phosphite. A picture tells 1,000 words, and here's a photo from this particular trial. You can see the foliage of the treated tree is very healthy, and the untreated trees have very thin canopies. Here's another photograph, and there's treated tree and untreated tree. I'm not a proponent of doing trunk injections with potassium phosphate. You have to be very careful within bark trees not to cause more injury than good, because you need to surface disinfect the areas where you're going to be drilling holes, and you have to also sterilize the drill bit to make sure that you do not introduce bacteria or fungi, which could then cause slime flux in those injected areas. So basal bark spray is probably the most effective and least damaging to the treatment to protect the tree from beech leaf disease. Trunk injection is a very inexpensive way to manage beech leaf disease, but it could have the consequences of harming the tree in the long term. So I'm favoring the use of a trunk spray, a bark spray, 2 applications per year to obtain a high enough concentration in the triaphosphite to protect the buds during the winter from damage by nematodes. It's really quite extraordinary for me to see foliar nematodes are among the most difficult pests to manage of all pests that we deal with, and to see that we have found effective treatments for protecting the health of forest trees from a foliar nematode is really quite extraordinary. Potassium phosphite is really the leader, in my opinion, because it's inexpensive. It can protect trees that are subject to reinfection of the buds, can be used at any sites, including landscapes and forest trees. It probably has a low environmental impact. It's not a restricted use pesticide, and it can be a do-it-yourself treatment with simple equipment. If you're doing the bark spray, all you really need is a small garden sprayer to do that kind of treatment. So my newest guidelines are to use a basal bark spray, dilute Reliant or a similar product that's registered for use for plant protection, to a 1-to-1 dilution. You wet the bark from the base of the trunk in the number of height in feet equal to the diameter of the trunk in inches. So if you have a 4-inch diameter tree, I would suggest that you really only need to spray to wet the bottom 4 feet of bark, and that might protect against phytotoxicity from salt damage. And then make 2 applications. Really, the best would be to go May and then again later in June. And then follow up with a foliar spray with the foliar spray rate. Follow the label carefully for the shaded foliage. But remember that the effectiveness of this treatment is going to be influenced by the condition of the foliage on the trees at the time of treatment. If the leaves are not functioning very well, then systemic products are not going to be accumulating in the foliage very efficiently. If you want to find more details about management of beech leaf disease, we have it posted both in English and in a Spanish translation at the Connecticut Agricultural Experiment Station website. So I'll take questions from the audience now. I don't know if you'd like to read